for joining us again for our weekly Wednesday webinar series. As always, we're going to give it just a little bit of time to allow the room to fill up. But happy Wednesday and welcome back. We are very happy to have you here. All right. Welcome. Thank you for joining. Right. Well, good afternoon, everyone. Thank you for joining us once again for our weekly Wednesday webinar series. Today's webinar is on a Canadian observational study for factor product utilization and health outcomes in patients with hemophilia on extended half-life concentrates. My name is also Mirtha, and I'm the Manager of Strategic Communications here at the National Hemophilia Foundation. At any point during the webinar, if you would like to ask a question, please use the Q&A feature at the bottom of your screen. We have NHF staff members monitoring these questions, which we will pose to our speaker after the presentation. This webinar is being recorded and live streamed and will be available to the community beginning on Friday, June 17th. So a quick hello to all of you watching on different platforms. I am joined today by Dr. Linda Sun. Thank you for taking the time to join us. I'll go ahead and turn it over to you to get started.
Thank you to the organizers for inviting me. It is my pleasure, and thank you to Dr. Valentino. My name is Linda Sun. I'm one of the adult hematologists at the University of Alberta Hospital, and I'm the medical director of our adult hemophilia treatment center here. I'm presenting on behalf of our study investigators on this Canadian observational study looking at factor product utilization and health outcomes for patients on extended half-life concentrates. So my relevant disclosure is that this study I will be describing is supported by an investigator-initiated grant by BioVeritiv, now Sanofi. So the past few decades have seen a major change in paradigm of hemophilia care. Looking at the timeline of the advances, ever since the initiation of prophylactic factor replacement, the first few decades have been spent in technological and improvement of the manufacturing process to improve really the safety of plasma-derived factor concentrates. And it's not until the 1990s where we had the first generation of recombinant factor products. The subsequent decade has seen generations of these recombinant factor products to further enhance the efficacy and safety. And it's not until just under 10 years ago where the first extended half-life factor products have become available. And since then, the treatment armamentarium have really exploded. There's a lot of options for patients and clinicians. Historically, most of us are familiar with prophylaxis with standard half-life clotting factors, and now we're faced with options of extended half-life factor products, non-factor replacement products, as well as the prospect of gene therapy. For today, we will focus more on the extended half-life product and our Canadian experience. The advent of extended half-life products, or EHL, has led to a major reduction in the treatment burden faced by patients. Imagine this is a treatment calendar of a young boy with hemophilia, and previously on a standard half-life factor products. Each of the circled days are the days where they have to do self-infusion and having to incorporate this in daily school, work, sports, and it can be quite difficult to juggle everything. And this is a sample calendar on the right-hand side of patients on extended half-life factor products. Because of the changes in treatment options, we're gradually shifting the goals we're trying to achieve for hemophilia treatment, and similarly with changing phases of patients with hemophilia. Initially, the introduction of prophylaxis, our goals were merely to reduce or minimize the number of bleeds and prevent long-term joint damage. And now, as more and more patients are on excellent prophylactic regimen with close to zero bleeds, we're becoming more ambitious and really trying to shift to the right side of this goals picture to enhance quality of life, to really engage people in day-to-day work, physical activity, normal living, and essentially try to normalize the life of a hemophilia patient and family to normal individuals. And as a Canadian, because our publicly funded healthcare system, always at the back of our mind as clinicians is also society costs and resources. To the forefront of people's mind may be the expensive cost of the factor products, but this is also juxtaposed with the cost savings of improved quality of life, reduced absenteeism, or loss of employment, things like this, as well as reduced emergency visits and hospitalizations. So just an introduction for patients who are less familiar, that there's a tender process for clotting factor concentrates in Canada. So factor concentrates are publicly funded by the Canadian Blood Services, or CBS, in all across Canada, except for the province of Quebec, where there's a separate organization called Hemo-Quebec, and it is dispensed by blood banks. So CBS, as well as Hemo-Quebec, independently conduct periodic requests for proposals called RFPs, every 2 or 3 years, typically, to decide which manufacturers or products are awarded contract. So as a result of this, because it's a public payer system, our availability of products may be a bit more limited compared to a market economy. We have an advisory committee external to the Canadian Blood Service that make recommendations, and based on scoring each product on pre-specified criteria ranging from effectiveness, safety, the reputation of the company, other values added. There's a long list of criteria, but ultimately also on which products offers the best value for money. As you can imagine, most of the factor products that are already approved by regulatory bodies like FDA or Health Canada, they all have excellent efficacy and effectiveness profile. So ultimately, small differences in cost or price makes a big impact on our availability of our products. And another thing that's rhetorical is what constitutes value in terms of value for money. Historically, we focus on more objective measures like number of bleeds, joint health, but now more and more there's an emphasis on a value that are more relevant to patients and families. So this is an evolving landscape of value-based healthcare. So as a background to our study, extended half-life factor products, recombinant factor 8 FC, also known as Eloctate, and recombinant factor 9 FC, Alprolix, first became available through the Canadian Blood Services in early 2016. For a period of over 2 years, these were the only available EHL products in Canada. And this is because of our tender process to be able to provide cost-effective therapies. Between 2016 to 2018, we only have one option for factor 8, extended half-life, and one for extended half-life factor 9. And then there is a new RFP process right before 2018 that led to the unavailability of these brands, just based on the new RFP process. So then patients have to face the need to switch to the alternative that were approved, which is a Dynovate or Rebenine. This is a small exception of people who are already on Eloctate for the purpose of immune tolerance induction in inhibitor patients, or people within 100 exposure days where the clinician or the patient do not want to switch early on to a different brand. But otherwise, there's very few limited options, as you can see. It's not until the most recent RFP, based on feedback from patients, families, and clinicians, they decided to offer more variety and options. So now, as of April this year, just a few months ago, we're fortunate to have 4 different extended half-life factor 8 options and 2 extended half-life factor 9 options. And emicizumab has only become available to patients without inhibitors as of fall last year. So we were quite excited about all the options we have now. But at the early stages, when the first extended half-life products became available in Canada, there were very scarce real-world data outside of clinical trials on how the availability of these products would impact factor utilization, clinicians' practice patterns, and patient-reported outcomes. And we also find similar data is lacking for patients on standard half-life factor 8 and factor 9 products as well. So because of these gaps, we conducted the Canadian Multi-Center Observational Study. It's a primary objective to describe the changes in annualized factor utilization of factor 8 FC and factor 9 FC over a 2-year period in men with moderate and severe hemophilia A and hemophilia B in Canada. Our secondary objectives is to describe annualized factor utilization of the standard half-life products over the same 2-year period as a background information, and also assess the impact of the FC products on clinical outcomes, but more importantly, a battery of patient-reported outcomes. and explore some of the clinicians' reasoning as to why they would switch someone or not, and their prescription patterns. This was initially designed as a prospective cohort study. As I'm going to show later, there are some logistic barriers in terms of enrollment, so we have later amended the protocol to include a retrospective cohort component as well. So the inclusion criteria for the prospective arm of the study is we intended to enroll consecutive patients with hemophilia over age 6 with either moderate or severe hemophilia A or B who do not have active inhibitors across 8 major Canadian sites, both adult and pediatric. And the enrollment occurred between April 2016 to June 2018. And patients were enrolled regardless of their treatment regimen, whether they were taking prophylaxis or on demand, whether they were on extended half-life products or standard half-life. And then the decision whether or not to switch someone to extended half-life early on at the time was solely based on shared decision-making between physicians and their patients, regardless of whether they participate in the observational study. So at the time of enrollment, we looked back at the infusion and bleeding records in the preceding 12 months. And we have also collected longitudinal data coinciding with their annual clinic visits at the 1 year and 2 year mark. We collected annualized factor utilization, bleeding rates, a battery of patient-reported outcome questionnaires, as well as surveys of clinician practice patterns. In addition, there's a 3-month data collection point of patient-reported outcomes, because we hypothesized this phenomenon of response shift may be seen, where with any improvement or changes in treatment, if there were any improvement in quality of life, mood, they're more likely to be noticeable early on, because once we get used to a new medication or winning the lottery, something like this, our internal expectations and value system have shifted or changed, so that 1 or 2 years later, the perceived differences may have been attenuated or decreased. The primary endpoint was intra-individual change in the factor utilization of the 12 months prior to switch to the 24-month period after the product switch. Secondary endpoints include the intra-individual changes in factor utilization in non-switchers. You'd think that the non-switchers who remain on the same product would have the same factor utilization, but the reason we wanted to collect this is during the study period, there's increased adoption of population pharmacokinetics and personalized prophylaxis. So we anticipate there may be changes in background factor utilization just because of changes in general treatment philosophies. We also collected annualized bleeding rate as well as annualized joint bleeding rate, the dose and dosing interval, both initially as well as with annual follow-up, the number of prophylactic infusions per year, physician practice patterns, and adverse events. We included a variety of age-appropriate patient-reported outcome measures, or PROMS, based on their validity, relevance, and burden of administration for our patients. For health-related quality of life, as a general overview, we included both a generic measure as well as disease-specific measure. The generic measure is SF36 because there's availability of population-based norms in the general Canadian population to compare with, and the disease-specific instruments we chose were Hemocool questionnaire for adults and chocolate for pediatrics. Recently, there is an international standard outcome set working group that developed through consensus a set of 10 patient-relevant health outcomes to try to harmonize longitudinal data collection of different studies, but also improve value-based healthcare. So that for future studies, the data collection is not so much focused just on bleeding or joint health or factor utilization, but with more incorporation of metrics or domains that may be more relevant to patients, but not historically measured, especially in the advent of all these novel therapy. So while our study was designed before the publication of this study, but for the proof of concept, we mapped our patient-reported outcome measures that kind of match to most of the domains of this framework. So on the left 2 columns are the sort of instruments we used, or subscales, that match the sort of described domains of different patient-relevant outcomes. And then on the right column are some of the recommended instruments by this international consensus working group based on their psychometric properties. So most of these were either from Hemocool or SF36 subscales, but for number of days lost or work productivity, we used a specific work productivity and absenteeism questionnaire For chronic pain, we used a chronic pain scale. It's a Likert scale from 0 to 10. And for physical function, we also included International Physical Activity Questionnaire. Treatment burden was assessed through the Treatment Satisfaction Questionnaire, abbreviated version. And for patient preference, rather than incorporating yet another questionnaire, just in consideration of convenience and burden for patients, we developed just a homegrown single question at the end of the study, in retrospect, looking back, would you have preferred to be on your current product? If not, why? So to see patient preference for a standard half-life or extended half-life product. So we used descriptive statistical analysis, and for our primary endpoint, Wilcoxon sign rank test was used to assess within individual changes in factor utilization and bleeding. For the sample size calculation, we estimated we need to recruit at least 25 patients who switched to recombinant factor 8 FC and 14 patients who switched to recombinant factor 9 FC to detect a statistically significant difference in the factor utilization. However, this was not reached in the prospective cohort study, unfortunately, because of delays in getting ethics approval of all centers and contracting. Some of the centers did not start their enrollment until 6 to 12 months after the first site opened. And we found out later that some large centers are early adopters. Rather than switching patients to extended half-life products at annual routine visits, some big centers switch a huge bolus of patients within the first 6 months when these are available. It just really shows, the impact of improved treatment burden for our patients. So at any rate, because some big sites are early adopters, they already enrolled patients and were not able to enroll them in the prospective study. So another kink of the study is that by 2017, it was announced through the RFP process that these brands, the recombinant factor 8 or factor 9 FC, would no longer be available in Canada. They're going to be replaced by a different EHL brand. So that also limited further recruitment. We did not make direct comparisons between switchers to extended half-life products versus non-switchers because of intrinsic differences in their baseline characteristics. For the evaluation of patient-reported outcomes, we didn't use formal statistical testing due to small numbers. Furthermore, a statistical significant difference does not equate clinically relevant difference in a change in quality of life. So for clinically meaningful changes, these were actually assessed by pre-established thresholds. So there were minimally important difference or MID thresholds previously established for SF36 and its subscales. And for hemoqual, there were previously established thresholds called responder definitions to denote, what magnitude of change would be clinically meaningful. So over a 2-year period, we had 158 patients who switched to one of the extended half-life products, including 93 patients switched to recombinant factor 8 FC and 32 who switched to recombinant factor 9 FC. As you can see, the vast majority are from the retrospective cohort. Pediatric population under age 18 constitute about half of switchers to recombinant factor 8 FC and about a quarter of the switchers to recombinant factor 9 FC. And the vast majority of patients, as expected, were on prophylactic replacement. Now, this table may be a bit busy, but I just wanted to point out, there are intrinsic differences of people who elected to switch to extended half-life products versus those who were content to remain on their standard half-life products. For instance, switchers to factor 8 FC tended to be younger, but they also have a more burden of disease, which kind of made the rationale for them to switch to something. So for instance, they have worse joint health. The median HJHS joint score was 27 in adults who switched to an extended half-life products compared to only 8 for non-switchers. Similarly, higher proportion of patients with target joints among switchers. And when looking at the baseline annualized bleeding rate in the 12 months preceding the study enrollment, the median ABR was 2 in adults who switched compared to 0 in non-switchers. Similar patterns we're seeing in switchers to factor 9 FC as well. So not surprisingly, we noted a significant reduction in the prescribed factor dosing per week. So for hemophilia A, there was a 14% reduction in the median prescribed weekly prophylactic dose from 99 unit per kilogram on a standard half-life factor 8 to 85 unit per kilogram on an extended half-life factor. The magnitude of reduction is less in adults, but both of these are statistically significant. In hemophilia B, on the other hand, the pattern is very similar between pediatric and adults, where there's about 44% reduction in the prescribed prophylactic dose from 100 unit per kilogram down to 56 unit per kilogram per week. However, when we looked at the actual factor utilization, which is based on patients' infusion diaries, these remained fairly stable in patients who switched from recombinant factor 8 to recombinant factor 8 FC. So for instance, looking at adults, the median annual factor utilization remained about 3,300 to 3,400 unit per kilogram per year without any difference before and after the switch. In pediatrics, there is a small drop of 7% reduction in the actual factor utilization, but this is not statistically significant. But I just want to point out how this contrasts with the calculated or predicted reduction in utilization based on the prescription. So in real life setting, based on the infusion diaries, there's not as much of a reduction in utilization. In contrast, we found a significant reduction in factor 9 utilization after patients switched from recombinant factor 9 to recombinant factor 9 FC, from 3,900 unit per kilogram per year on a standard half-life product to 3,300 unit per kilogram per year. And this is very similar for both adults and pediatric population. Only the adult population showed a statistically significant change, but the numbers are small for the pediatrics. And again, I want to contrast that for both adults and pediatrics, the actual utilization, we observed a 15 to 16% reduction after switching to extended half-life product, and in contrast to a predicted 44% reduction in utilization based on the prescribed dose. We were very reassured to see that the bleeding rates remained low for patients who were on prophylaxis who switched from a standard half-life to extended half-life product. So for adults, the median ABR was low to even start with, so about 1, without any changes before and after. For pediatrics, there's a statistically significant reduction in the annualized bleeding rate from 1 on a standard half-life product to 0.5. Even though it's statistically significant, the magnitude reduction is fairly small. Similar patterns is seen in switchers to recombinant factor 9 FC. So in adults, the median annualized bleeding rate remains stable. In pediatrics, there's a similar magnitude of reduction from 1 to 0.5, although this is not statistically significant. Numbers are also smaller in this group. We also did similar analysis looking at the rates of annualized joint bleeding, as well as the proportion of patients who had 0 bleeds before and after switch, and we did not note any significant changes in those. Now, looking at the prophylactic regimen in patients with hemophilia A, so for both adults and the pediatric patients, the blue bar represent what prescription they were on prior to switching to long-acting half-life factor product. And most patients are on every other day or 3 times a week between pediatric and adults. However, when they were switched to extended half-life factor 8, we noticed some more difference between adult and pediatric clinicians. So for pediatrics, it seems more homogeneous. So the vast majority of patients got every 3 days or twice a week. In adults, it's more heterogeneous. About a third of the patients actually remained on a more frequent prophylactic regimen, so every other day or 3 times a week. And then about another third were on twice a week, and yet a quarter of the patients were able to extend it further into once a week. So looking at patients with hemophilia B, while on the standard half-life factor 9 products, looking at the blue bars, the most of the patients are split between twice a week to every 2 to 3 days in both pediatrics and adults. Again, in pediatric practitioners, the prescription pattern seems to be a bit more homogeneous, where all patients were switched to once a week prophylaxis. In adults, again, a small proportion of patients were maintained on a more frequent prophylaxis, so twice a week, just as they were when they were on standard half-life, and then 61% on once a week, and a small proportion of patients were able to extend it even further every 10 to 14 days. So these pie charts just showed some of the changes patients experienced before and after switch. It may look a bit busy, but I just want to highlight the blue slice of the pie, where about a quarter of patients who switched to either recombinant factor 8 FC or the recombinant factor 9 FC were maintained on the exact same regimen, like the same factor dose and dosing frequency before and after. Presumably this intent to raise the factor 12 level to reduce the rate of breakthrough bleeding, microbleeding, or empower more physical activities and exercises. When we surveyed clinicians as to what are some of the common reasons for switching, and clinicians can choose multiple options, like up to 3 choices, the most predominant reasons for switching are similar between adults and pediatrics, which is being able to empower less number of infusions, so convenience for the patient, respect for patient preference for this product, as well as quality of life. This is followed by frequent bleeds. The other reasons are fairly rare, so about only 10% or less of the patients were switched predominantly due to poor venous access, a documented short half-life on their standard half-life product, the desire to do more sports or physical activities, etc. But I do want to point out, interestingly, there's some differences we observed in the rationale between adults and pediatric treaters, as in about over 80% of pediatric patients were switched because of improvement in quality of life is being prioritized, whereas only 20% of adult treaters switch their patients to an extended half-life product is because of quality of life. And this reflects just in a publicly funded healthcare system, these were more expensive than the standard half-life product, especially being a new thing. A lot of adult treaters just, to be cost conscientious, may not have felt improvement in quality of life as a major motivator for a more expensive therapy. In contrast, over 25% of adult treaters switch their patient because of frequent bleeds, and frequent bleeds is less common in pediatric population as a reason for switching. When we surveyed the clinicians, and again, this is earlier on before population PK became more dominant, about 42% of individuals were being planned to have population PK or a factor 12 activity measured. And we found that adjustment in prophylactic dose or dosing frequency in the first 1 to 2 years is fairly common, more so in pediatrics than adults. We did not observe any significant adverse events such as inhibitor development, allergies or hypersensitivity reactions or other serious adverse events. And reassuringly, most of the patients elected to remain on the extended half-life products throughout the study. They were quite content. Only 4 patients elected to switch back to their previous standard half-life product because of their perception of increased bleeding or bruising tendency, and 1 person had frequent headaches, which they thought may be related. Now, shifting gears a little bit, looking at patient-reported outcomes. So the number of patients are smaller. So 16 patients who switched to recombinant factor 8 FC and 9 patients who switched to recombinant factor 9 FC completed the patient-reported outcome longitudinally, as well as a number of non-switchers as well. Just wanted to point out that the switchers, who completed the patient-reported outcome questionnaires compared to non-switchers were older. They tend to have a higher baseline bleeding rate, so ABR of 3 compared to 0 to 2 among non-switchers. And they also have lower quality of life at baseline at the time of study enrollment. So HemoQoL is the overall health-related quality of life questionnaire rated from 0 to 100. The lower the score denotes better quality of life. So switchers have a higher score, meaning like a lower quality of life compared to non-switchers at the beginning. So this table kind of abbreviated shows some of the key, the overall score, as well as some of the key subscales of the HemoQoL questionnaire. We sort of selected to highlight here these subscales because these were the ones have established responder definitions. So just as a reminder, responder definition is that threshold that denotes what's clinically meaningful or clinically significant. So it may look a little bit busy, but I just wanted to highlight that most of the improvement, which is in the direction of a negative or a reduction, is seen in the domain of physical health, less so in other domains. And the other thing I wanted to point out is that patients who switched to recombinant factor 8 FC tended to have smaller magnitude of improvement compared to people who switch to recombinant factor 9 FC. For instance, people who switch to recombinant factor 9 FC, there's a mean reduction of the total score by 5. And so in the direction of improvement, compared to only a reduction by 2.3 for switchers to recombinant factor 8 FC. And correspondingly, only 12% of switchers to recombinant factor 8 FC achieved that clinically meaningful improvement, like met this threshold of -7 in the total score at 3 months, compared to 29% of switchers to recombinant factor 9 FC who met this clinically meaningful improvement. And the other thing that's really interesting, which kind of met our hypothesis, is that the initial improvement seen early at 3 months are kind of disappeared or attenuated by 24 months. So for instance, the total score, the improvement was -5 at 3 months, but close to 0 at 24 months, which really reflects that internal shift in expectations. And these are the SF36 results. So these are radar plots, which show among the 8 different domains of the SF36, what are the scores. So SF36, similar to hemocool, also ranges of a score from 0 to 100. But the direction is opposite. The higher the score means the better quality of life. So among switchers to recombinant factor 9 FC, we noticed improvement in selected SF36 domains. So for instance, at baseline, which is denoted by the black colored contour, compared to the gray lines, which is at 3 months, or the red contour at 24 months, there is improvement or kind of an outward expansion of certain domains. But this is not really seen in people who switch to recombinant factor 8 FC. But overall, about half of switchers to either recombinant factor 9 FC or recombinant factor 8 FC met the minimal important difference of a clinical improvement in health-related quality of life based on the physical component scale of SF36 as well as the mental component scale. We measured quite a large number of patient-reported outcome measures, but just for the interest of time, just want to highlight a few select ones. In terms of chronic pain, we noted an improvement in chronic pain in patients who switched to recombinant factor 9 FC, but not recombinant factor 8 FC. For instance, the chronic pain scale from 0 to 10, it improved from a baseline of 5 out of 10 chronic pain in the preceding month to 2 out of 10 at 3 months. And this coincided with also an improvement in the SF36 subscale of bodily pain. And we found that about 56% of switchers from recombinant factor 9 FC was able to meet the minimal important difference threshold of SF36 bodily pain at both 3 months and 24 months. In mental health, again, over half of switchers were able to meet the minimal important difference in the mental health subscale of SF36. However, this is only at 3 months. It's not seen at 24 months. In terms of work and classroom absenteeism and impairment, the baseline level of absenteeism or impairment is fairly low. So there's the ceiling effect. We did not see any further changes at 3 months or 24 months. This certainly may reflect selection bias, as people who are quite sick, needing lots of emergency visits and missing work, they're less likely to enroll in this observational study and do all these longitudinal questionnaires. And our homegrown patient preference questionnaire at the end of the study, when we asked in retrospect, would you have preferred to be on this product you're on, either for people who are on EHL or standard half-life products, all but one switcher indicated that absolutely they would prefer, their decision to switch to EHL over standard half-life. So overall, we have shown that the switch from standard half-life to extended half-life products in Canada was associated with a significant reduction in the prescribed weekly prophylactic dose. However, when we look at the actual factor utilization based on patient infusion diaries, the significant reduction in factor utilization has a smaller magnitude and also only observed in patients who switch to recombinant factor 9 FC, but not in patients who switch to recombinant factor 8 FC. It's interesting that we observe this discrepancy between the expected factor utilization, which is based on the prescribed dosing, compared to the observed changes, which is based on the infusion diaries. And there's a few possible explanations because this switch to extended half-life product, we do expect people to improve on their adherence to their prescribed prophylaxis. So this may have narrowed essentially the gap in factor utilization when they were on standard half-life versus extended half-life. And also the study took place in an era where there's increased adoption of activity-based prophylaxis, where in addition to routine prophylaxis, people give them additional factor concentrates before high-risk activities or just routine sports, and also population PK-based personalized prophylaxis. So this may have attenuated, again, the expected reduction in the factor utilization with the switch. And we also want to point out that, when these extended half-life products first become available, there's a lot of concern of increased switch and increased cost on the system level in a publicly funded healthcare system. So perhaps more relevant to the payers is what's a factor utilization before and after, but this is not the most patient relevant measure. And I also want to point out that about a quarter of patients were actually maintained on the same dose and dosing frequency before and after the switch to extended half-life products, not to save costs, but really to enhance that factor trough level and reduce number of bleeds, microbleeds, and potentially improve future joint health and chronic pain and all these other benefits. It's reassuring that we found the overall bleeding rate remained low both before and after switch, which really just is reassuring that overall, even with standard half-life, most Canadians are doing fairly well on a good prophylaxis regimen. We demonstrated a statistically significant reduction in the median annualized bleeding rate only in pediatric patients who switched to recombinant factor AFC. And this reflects a ceiling effect, because when people are already doing very well in a developed world with good access to universal healthcare and publicly funded factors, it may be difficult to demonstrate either a statistically significant reduction in bleeding or a clinically relevant reduction. I suppose looking at the proportion of patients with zero bleeds may be a more sensitive measure when the baseline ABR rate is already low, although in our study, we didn't show a significant difference in the proportion of patients with zero bleeds either. The other thing is the study may be underpowered due to the sample size. And finally, because 3/4 of the patients actually had a reduced frequency of prophylaxis, only a quarter of patients maintain the same dose and dosing frequency, you wouldn't necessarily expect the ABR to improve, but rather what you would expect is just a greater impact on treatment burden, patient satisfaction, adherence, and those other measures, which we try to capture in our patient-reported outcome part of the study. In terms of patient-reported outcomes, we demonstrated a small improvement in the overall health-related quality of life as measured by the HemoQual total score in switchers to extended half-life products, specifically more so in recombinant factor IX FC, where over a quarter of patients met the pre-specified cutoff for a clinically meaningful improvement in quality of life. Improvement, as I mentioned before, is seen more so in hemophilia B patients and hemophilia A, and in a variety of different domains, ranging from physical health to mental health and chronic pain. However, the responsiveness of the current tools or these instruments and ceiling effect may limit any perceived impact of novel therapy, because in the developed world, people are often already doing fairly well. That is a good baseline. So hence, it's important to really take into consideration the ceiling effect when evaluating patient-reported outcome measures following switches to novel therapies, either extended half-life products or non-factor replacement products. Because I want to highlight that most of these measures, instruments were developed in the era of standard half-life products. So they're more likely to demonstrate an improvement or responsiveness when patients switch from on-demand treatment to prophylaxis. They were not designed to assess if the switch from standard half-life to extended half-life or to a non-factor based treatment regimen is going to lead to a significant improvement. The ceiling effect of the current instrument have been highlighted by a number of other studies, including limited ceiling effect seen in chocolate, the hemophilia specific quality of life questionnaire for kids, as well as HemoStat, which is a satisfaction questionnaire. As a result, there are ongoing efforts to update and revise to create new versions of these patient-reported outcome measures to be more in line with the current era. For instance, the group at the Hospital for Sick Children in Canada has established chocolate version 3.0. And then there's ongoing efforts for some of the other instruments as well. But what is perhaps more powerful to our study team is at the end of the study, when patient was asked that single question, would they have preferred the standard half-life or extended half-life? All but one patient indicated preference for extended half-life product. So to us, it's a very compelling evidence argument that the current PROMS, however sophisticated they may be, they may not be responsive to some of the metrics that are more relevant and important to our patients. I just also wanted to contrast our real world experience in Canada compared to some other countries. So just to highlight, our Canadian study that looked at the period from 2016 to 2018 showed stable factor VIII utilization after switching to EHL and reduced utilization in patients who switched to factor IX EHL products. In the US, Croteau and colleagues looked at an 18-month period similar to our study period, actually, of the Thrombosis and Hemostasis Network affiliated hemophilia treatment centers, and they showed a reduction in factor utilization after switching from standard half-life to extended half-life for both factor VIII and IX. And interestingly, if you look at the annualized factor utilization, like in terms of unit per kilogram per year, our Canadian, especially in the adult population, in terms of the factor consumption is markedly lower, and especially in hemophilia A patients, and kind of and similarly, in some of the factor IX utilization as well, it kind of reflects historically a lot of our clinicians are more cost conscientious, but this pattern is certainly changing, especially with the advent of emicizumab and gene therapy. We may be aiming at higher factor XII levels than we had been historically. I also wanted to point out the Australian experience in their first 6 months of EHL availability. It kind of echoes our study where the actual versus expected change in factor utilization based on infusion diaries versus based on the prescribed dose is quite different. So similar to our study, even though there is a expected reduction of factor IX by 26 units per kilogram per week when switched to a factor IX extended half-life product, but the actual observed reduction is quite modest. And similarly, while they expected a reduction in factor VIII utilization after switching to EHL, the actual actually has a small increase. Of course, this is early experience, just 6 months, and while clinicians are still getting used to it, but just kind of highlights this discrepancy, which we also showed in our study as well. So some of the strengths of our study, it's very robust 2-year observation period across the major Canadian adult and pediatric hemophilia treatment centers. We purposefully allowed for this longer observation period to allow time for dosing stabilization and any changes in a clinician treatment patterns. We have, in addition to the traditional measures of bleeding rates and factor utilization, included a large variety of patient-relevant outcomes to really try to get a sense of the other benefits in addition to minimization of bleeding. And we also elected to use within individual comparisons before and after the switch to reduce the risk of confounding, as will be seen in between group comparisons between switchers or non-switchers. Now, the main limitations of the study is that the most data were actually from the retrospective cohort component due to enrollment issues, which really reflects real life struggles in terms of delays in ethics and contracting in the current era, but also the constraints by our RFP process, where these products, even though we got investigator-initiated grant funding from the manufacturer, this product is no longer offered in our setting. One of the both benefit and limitation of the study is that we chose the actual factor utilization rather than the calculate utilization as a primary endpoint, but there is a risk of differential degree of incompleteness of infusion records because in the 12 months prior to study enrollment, a lot of patients were still using paper records and mailing them in and faxing them, so leading to some incomplete records. Throughout the study, there's increased adoption of the Canadian Bleeding Disorder Registry across center, where this is a national-wide registry where patients log their infusion records and bleeding. So this may create a risk, a bias, towards a null, where we may have underestimated the annualized factor utilization when patients were on standard half-life products before the switch. And the other limitation is, while we collected for which patients did physicians collect PK data, we didn't actually get the details on the PK profile and how it may have affected the factor utilization. So with that, I would like to acknowledge all the patients and families who contribute to our study, our steering committee members, and all the participating sites and study staff across our country. And I would also like to thank you for your attention and for the organizers for having me here. So I'm open for any questions and discussions.
That was wonderful. I do have a couple questions for you. Let me go ahead and get that pulled up. Okay, so my first one is, do patients in Canada, oh, I'm sorry, before I actually begin, if anyone has any additional questions, please make sure you throw them in the Q&A feature or in the chat so we can get them asked. And we are still monitoring YouTube, LinkedIn, and Facebook if you're viewing from there. So our first question, do patients in Canada have a problem changing from one product to another every 2 years or as long as the tender lasts? And have they shared their personal experiences or concerns?
Yeah, so that's an excellent question. I think it really varies from patient to patient. In the early days, because this has been going on for a while, there's lots of concern of these forced nationwide switches where half of the country have to switch from brand A to brand B in terms of potential inhibitor risk. But since then, there has been a large number of studies from other publicly funded countries, not only from Canada, but also UK, where there does not seem to be as much concern. So most of the patients are not concerned about the safety or inhibitor development aspect. But what we find is just that burden or inconvenience, especially now population PK or pharmacokinetics are increasingly done. So oftentimes we do PK before and after, on different brands, that leads to extra clinic visits, lab visits, getting familiar with a new brand and, the system. So because of that, our most recent RFP process, from very loud and clear messages from our patient communities, as well as treaters and families, that we want more choices. We don't want to force people to just, have to switch from this brand of extended half-life, like Eloctate, to a Dinovate, which took place, so that fewer and fewer patients would need to switch in subsequent tenders. Yeah, but that is absolutely a lot of feedback from our patients.
Thank you. Does the relatively small reduction in SHL factor usage equal cost savings since EHL products are more expensive?
Yeah, so even though based on the prescribed dosing, we would expect potentially cost neutral in real world experience, at least from what we gathered, but also the Australian data, there may or may not be much reduction in factor utilization. So it may have been more expensive. We thought to include a health economic analysis, but did not pan out. But since then, the price, thanks to our RFP process, it has led to significant reductions in the cost of EHL over the past few years. So I think in the current era, it's fairly cost neutral. So because of that, we have really shifted before from just one EHL per hemophilia group, A or B, to now 4 options for people with hemophilia A, and especially this availability of emicizumab. So that it's really changing. So I think back then in the study period, it probably did not lead to cost savings and maybe more expensive, but we didn't do the particular calculations. And now it's quite different.
Wonderful, thank you. So another question, what happened between 2020 and 2022? Were any EHLs still part of the tender?
Yeah, so things got delayed from the pandemic. There was supposed to be, so the tender was initially for 2 years. And then they kind of just got extended. So the most recent tender did not take place until last year, and the initiation date is April this year. But since then, there had been some slight changes where it's more brand specific. So Jivi, because Kovaltry, the manufacturer, is already available as a standard half-life, and they introduced Jivi, and I believe they made it fairly cost neutral. So then patients who were already on Kovaltry, they were allowed to shift to Jivi. But for people on other standard half-life factor, there's not that equivalency. So there are some slight opportunities for people to switch. And similarly for Eloctate, even though it became not available for selected patients, we have to fill out special forms. For example, taking it for ITI, as I mentioned, or the first 100 exposure days, we can make special requests. But for the most part, it's fairly restricted. So those 2 years, we just extended from the previous RFP for the most part.
Great, thank you. Will there be any studies that include women?
That is an excellent question. Yeah, historically, just due to some intrinsic sexism and culture in medicine, a lot of these studies we only included men, but certainly there are women with potentially moderate and severe hemophilia as well. So that is, we did not take that into consideration when we wrote the protocol way back in, at the beginning, but it's certainly something that will be considered in future study designs.
Great. So I have another one for you, and I'm unsure if it's PROM or PROM for the word, but the question is, I saw a lot of PROMS in the adult arm of the study, but in the pediatric study, there was only one tool. Is that because of the lack of validated tools in various categories to measure pediatric patient-reported outcomes?
Yeah, so that's an excellent question. Yeah, so for pediatrics, maybe the table is a bit confusing. I was trying to merge some of the cells, so it didn't show up properly. So the pediatrics also had SF36, as well as the hemophilia-specific quality of life tool, HemoQool, and they also did the chronic pain scale, except instead of numeric 0 to 10 is those phases, a visual analog scale. But you're right, there is absent of validated tools for some of the other things like treatment satisfaction questionnaire, the IPAC, International Physical Activity Questionnaire. Yeah, we didn't present the results for the chocolate just because as part of the study protocol, I didn't mention just the details and the method. We only reported results where there's at least 5 individuals for each metric with matched results pre and post. As pediatrics, sometimes there's dropouts where people filled out the questionnaire before, but no subsequent ones. So we didn't actually report the chocolate results in our final manuscript.
Great, thank you. So what were your greatest challenges in your research?
I think definitely the logistic challenges in terms of just increasing struggles of getting timely research ethics board approval, contracting between countries, but also this investigator-initiated grant dealing with industry partners as well. So there has been lots of back and forth that led to the delay of initiation of the study in terms of sample size. So with our sample size calculation, they obviously wanted to revise our protocol to mandate a higher sample size. So there's lots of back and forth in terms of just the details of the protocol that led to kind of delays. So by the time it opened in all the centers, including big centers, a lot of patients were switched already. Yeah, and also in our climate, the logistic barrier of the RFP process. So after this, we had some other products like extended half-life products, but a lot of our investigators were a bit more, we felt bitten or wounded by this experience. We're less likely to start, at least with industry partners, a new investigator-initiated grant with a new like a EHL product, because we're like, we don't know how long it's going to be here for before they become no longer available. But ultimately, I think lesson learned is that instead of brand specific products, like it would make sense. At the time, this was the only brand. So we reached out to the manufacturer for their funding for this product. But looking forward, it would make sense to be more inclusive, not focusing on just emicizumab or just one brand of EHL, but really looking at how all these options compare and contrast with each other.
Wonderful. Do you have any plans to build on your findings over the next few years?
Our center ourselves, we're not, to build on our findings.
Yeah, like I guess continuing your research.
Yeah. I think one potential interest is development of, more relevant patient-reported outcome measures to the current era because of the previous ones were outdated. But we find one of our colleague groups in the sick kids, as I mentioned, they developed novel patient-reported outcomes, both the chocolate 3.0, but as well as more treatment burden-related questionnaires to try to be more responsive to a small improvement in quality of life with a switch to, say, emicizumab or the novel therapy. In our adult group, we find there's lots of burnout the last few years, just from patients, the pandemic and everything. So we thought about, collaborating with that group to do more adult patient-reported outcome measures, but there's not lots of enthusiasm among the adult centers right now. People are just putting out fire from all the pandemic effects.
Fantastic, thank you. That is all the questions I have, but Len, do you have any that you would like to ask her during our last few minutes?
I think, Linda, you've addressed the questions that I had. I really think it's critically important so that we understand how best to optimize the use of the products that we have available to us to treat people with hemophilia. So I applaud your research and the effort that you went through, because I know this wasn't an easy study to be able to execute. So thank you very much.
Thank you again, and thank you for the excellent questions from everyone.
Yes, wonderful. Well, thank you so much, Dr. Linda Sun, for taking the time to join us this afternoon. As always, we appreciate your time and your expertise. I'd also like to thank each one of our viewers for joining us. Please note that this recorded webinar will be available on Friday, June 17th at hemophilia.org under the Events tab with all of our archived webinars. Also available in the Events tab is our upcoming schedule of our weekly Wednesday webinar series. Thank you again for joining us this afternoon, and have a wonderful week.
Thank you.